So today we have Wietske van der Zwaag here, and we're excited that she will talk about mesoscale MRI and fMRI in the cerebellum. Please.
Yeah, so thanks a lot for the invitation, Renzo. Also, thanks for giving me this title. I have to say this is actually what I would love to be talking about, but it's a little a little bit off into the future at the moment. So this is a talk not so much about mesoscale fMRI in the cerebellum, but on how we go there and what is my what are my thoughts on this process. So this is our cerebellum. This is actually my cerebellum, highlighted in light gray, just to show you that it's pretty and where it's sitting in the brain. And it is, to a large extent, a little bit out of reach. We do a lot of, in our neuroscience daily everything, we do a lot of focusing on the cerebrum because it's easier, it's accessible, you fit super well into the Nova 8-channel head coil, and life is a lot easier when you stay inside this red box. But this is silly because the cerebellum is super relevant. It's an integral part of the brain, and it's involved in a lot of neurological and psychiatric diseases. And if we want to understand from a network point of view what these diseases do, what happens, where it goes wrong, then you cannot avoid looking at the cerebellum. So I think we have no choice. But it has this very intricate structure, which kind of just makes life very complicated. And as an MRI person, I think that means that the MRI people should do better. So this is what I set myself to do as a challenge. So what is it that we're actually looking at? The human cerebellum has, it means little brain, and that is exactly what it is. It has this white matter structure, which is a bit tree-like. So some of this, it's also sometimes referred to as a tree of life, which contains deep gray matter nuclei. You can see the dentate here. And it's covered in its own gray matter cortex. This tree-like structure you can divide up into lobes, which is the largest division, lobules, which is the red thing I highlighted here, and folia. And within each of these bands, you get 3 different cell layers. So this is the mesoscale feature, if you want, that we most want to access, because this is where the computations happen, where we can say something about maybe forwards and backwards processes. So we know what we're aiming for. We can just about almost maybe see it. So if we look at that layer structure, then we know, well, all of you in this call are very well aware of this diagram. Our cerebral cortex has 6 cell layers, but we often divide it into a sort of a deep, middle, superficial layer. And we have some idea of what happens where there's input, where there's output or feedback. And then, again, we see a lot of calls, a lot of talks in this lecture thread where we get these sorts of plots, right? We divide up the cortex into different depths. We can plot our responses as a function of cortical depth. And then we can look at different ROIs, and we always see this rise towards the surface. And this is a graph from Josh, but there are many. So how would it look in the cerebellum? If we could do this, what would we look at? So we know that in this tiny little folded up brain, there are these three different depths, which also have each their own function. There are different parts in the cerebrocerebellar feedback loop, and we know from neuroscience, just sticking in electrodes and recording, what these cell layers are actually doing. And if you take all of that surface and flatten it, then in the other dimension you get a 2D space, much like the cerebral cortex. And then we see that there is sort of a parallel stripes. So they're not cortical columns, as you see in the visual system, but it's more of a stripe-like pattern. And these stripes have their own function. They're primarily characterized by difference in protein expressions, which we know humans also have, but these proteins, the way they're organized, they are related to function in that things like, so super basic, the flexion and extension of a forepaw are controlled from different stripes, different parallel regions. Both active during movement of a forepaw, but for different muscle groups. So this is a two-dimensional space, or three-dimensional space then, where you have a surface with stripes and a depth where we can talk about the network, much like how we think about cortical layers and columns in the cerebral cortex. And what does that network then look like? So I just wanted to talk you through this. See it as a roadmap, where these tracts go. So there's information coming initially from the spinal cord, which gets sent to the inferior olive, and there's a climbing fiber which projects to the superficial layer in the cerebellar cortex, which then talks to the Purkinje cell layer, which is the middle, and from that middle layer, all of the output goes to the dentate nucleus. So that's like the output gate, if you want, which sends signals up into the large brain through the red nucleus and the thalamus. And then here, the example is always the motor cortex, but it doesn't have to be the motor cortex. It could be anywhere in the brain. And then feedback goes back through the pons and then into the deep layers of the cerebellar cortex. So having this distinction between the two, between the different depths, can tell you something about what happened, like where in the loop this signal is coming from. This is all assuming that we can see these different depths and that we can do imaging, which almost, we're nearly there. So I keep this image on my desktop actually, to remind myself of what it is that we should be looking at. This is a postmortem brain from Marty Serino, which they took just a cerebellum, that's why there's no stuff around it, stuck it in a 9.4 and then scanned for, 36 hours or so. And they get these 0.2 isotropic, really well-defined images of a human cerebellum, a complete human cerebellum. And even without making any efforts in segmentations or anything else, you can look at this and you can tell that there's a cortex and that there are different layers inside this cortex and that these layers have different properties in terms of folding and thickness, depending on where exactly in the cerebellum you are. Now, sadly, most of our images look like this, certainly the functional ones. And you know that this is, or you can guess that this is a cerebellum. I know that it's a cerebellum. But when I first copy-pasted this into my PowerPoint, it said man in a cave, right? So it's not really where we want to be. But it is where most of our functional imaging is. If you look at the structure, we do a bit better. That'd be a 0.8. It's kind of, I think that's a standard resolution for 7T. It's not even particularly ambitious. If you try a little harder, you get to a 0.4. You start to see that there is actually this structure that we're looking for, this tree-like thing with that you have details and fine features that we might be interested in. And if you push beyond that, and this isn't even an isotropic resolution image, this 0.2 in plane, then things just get a little blurry and noisy and it takes too long to acquire in vivo, right? So we want to go somehow from this right bottom corner to the left and then stay in vivo. Partly also because a lot of what we take out of our images at the moment is just not realistic. So we have segmentation tools which describe the cerebellar cortex as something like this, which is if you try a bit harder and you get high resolution images, then you see that there's a lot of white matter actually going into these gray matter things. So if you want to do anything clinically relevant and say something about how thick or how much gray matter there is, or whether people are maybe losing mass in certain parts of their brain, then you need something much closer to this than to this. And then again, I'm an MR person, so my approach to this would be to get more SNR, because life is better with more SNR. And sadly, not all of our coils are equally well covering the cerebellum. So I thought if we start there, we just look at hardware. If we go close, right, we take a surface coil, we make it small, we make it appropriately sized for the cerebellum. Then we get these sort of patches, and this same coil has also been used for surface coil imaging in a visual system. So we just moved everything down, moved everything closer. And then you see that there is indeed much more SNR. So we can do this. It's not a super complicated coil to build either. But as soon as you do this, then people are like, oh, you get much more signal. That's great. Can I look at connections to the motor cortex? Can I look at connections to the frontal cortex? And the answer is no. So this works well, provided that you have a research question which is purely cerebellar. And I just showed you that network. It's very hard to think of anything in isolation. We do need to cover the entire brain. So sadly, we kind of abandoned the coil and then started looking at PTX instead. First using RF shimming, and this is not a cerebellum, it's not even a brain, but the idea is the same. You've probably seen this before. So if you look at any part of the human body really, at 7T or higher field, you get these black holes where the signal's just cancelled out because of the interaction between the transmit coils. If you change only the phases, you can do much better. You get signal throughout. And if we want to, because it's so simple, only changing the phase, you can do this easily, and you can make this reusable across subjects. You can make it universal just by our first attempt at doing this for a cerebellum is we just took the median phase offset across not even a very big group, 8 people, and then used that to generate cerebellar images, and you get way better results than in a quadrature mode. So it's cheap once you have the PTX, and it's as easy to use as any other scan. And what works then for, and this SNR, and that's important, right? So anytime you gain in SNR, you also gain in functional sensitivity. So this whole idea of not seeing responses or not seeing cerebellar involvement, you can fix that by just making sure that there is enough signal to be found in the cerebellum. So you can improve on this by doing advanced pulses, or we've used KT points a lot, but the idea is the same. So you use the PTX system to make sure that there is actually signal throughout the brain. A much less affordable option would be to increase the field strength. So there's people with very fancy scanners like looking at this, but so this is where we can really make the increase in SNR count. If you change, not like in this case, we changed nothing. We took our standard 0.4 anatomy from the 7T and went to Maastricht, which is about 2 hours on the train from here. Ran the same protocol in the same volunteers in which we'd previously scanned at 7T. And then again, without doing much in terms of analysis, you look at these images and you can tell that this one's more grainy than that one, right? And then that very quickly also translates into better morphometric measures. So if you do the segmentation, there's more iffy bits, more noise, more structures that shouldn't be there. There's even like a bit of like, I don't know how I would call this, but thicker branches in a completely not biologically relevant way. Right? The asymmetry here just isn't actually there in this brain. Well, I hope, but I assume that. So when you do the segmentation of the same thing on the 9.4, then you get a much more balanced image. So there is a point in looking for more SNR, and I don't think we've quite finished in eating up all the SNR that we can possibly have. But there's more. So once you get this super high resolution images, or actually as soon as you go below the 0.5 millimeter or so, then you also want to do something in terms of motion correction. And the way we did this is by using the FAT navigators, where you acquire a FAT image every 5 seconds or so, update the gradient system in a prospective manner, and then make sure that you kind of follow your subject through time. Which is not super efficient. The fact that you have to insert the navigators works for some sequences. So for the MP2 range, for example, it doesn't cost time, but you still have to insert something and then analyze that and do an update. So it's not maybe the best way of doing things, but it does help a lot. You get a lot more definition in people that are reasonably well behaved. And once you have it up and running, it's pretty user-friendly. It just makes the scan longer. So if you take all these things together, then we get these sort of images, which are unfortunately not yet the resolution of the post-mortem images, but they're still pretty good. You can see the actual individual folia, which is much better than what you can do at in a 1mm scan. You can see that they pass through. You get the idea of a concept of a sheet, right? So there's a cortical sheet which is being folded around all these white matter trees. What you also see is that this particular scan is just a small part of the cerebellum, and because of the anisotropic voxels, we can do this, but the validity of the segmentation is then also only limited over a small portion of the cortex. So this is I would say it was a very big step in the right direction. We can take more steps. But what you can also see is that once you've gained this, that when you look at not so much the magnitude of these gradient echoes, but the phase, that we start to see the actual layers themselves. So they're here, and is a white matter branch, and next to that is a granular layer. So where we assume the very bright area, where the granular layer would be expected, and then a less bright gray matter area. So now we don't see three layers, right? But we see that on top of the white matter there are two separate layers, which we can actually distinguish. And if you look at consecutive slices, then you see that they extend through the slices as well. So we know that these are the same as in a rat brain, which you can more easily scan at sufficiently high resolution. So we can, for once, do something which is a little bit beyond what we can do in the cerebral cortex. Because this granular layer is so pronounced, we can see it in the phase images. And actually, maybe once we get there, if we have sufficient resolution throughout, we could segment the different layers out of these structural images. It should be proper mesoscale. So Nikos, who's done a lot of work on Nicolas Plirovlus, I should say, I'm going to use his full name. He's done a lot of work on this, also in terms of making the images not just good images, but actually usable and quantifiable and maybe clinically relevant as well. So here he set out to acquire to segment three different sets of images. First, the in vivo, which are the ones that we acquired, then a post-mortem MR, the publicly available MR dataset, and a 3D reconstruction of the big brain for the histology, so that we can start to link what is true, right? Where do we see these layers? Is what we see in the MRI, is that actually related to the biology? Because if you look at stained sections, then we can really probe where these layers are. Right? We have in three different stains of this one slice, you see that there's very clearly a granular layer, this molecular cell layer at the top, and you see the white matter branching. Note that also here, you don't see the Purkinje cell layer, it's just too thin. So that requires another big step in terms of in terms of the resolution, or maybe in terms of, I don't know, specificity of the images or something. But for now, we're trying to get a handle on these layers and see if we can say something about this, and also then characterize how these layers behave. So, for example, if you look at the cortical thickness, so the cerebellar cortical thickness, that is much more related to the granular cell layer because of how things are packed. So, depending on the curvature, the overall thickness follows what the granular layer seems to do. So maybe if we have a really good segmentation of the cortex, then we could also use that as a marker for what the granular cell layer is doing underneath. And then there are other things you can also target. So once you have the segmentation at this quality, then you can look at things like how does the T1 behave across the surface? How does the T1 behave as a function of curvature? How does the T1 vary across the different lobules, which is because the different lobules have different functions? How can we relate any of these to what we know that we can easily access in vivo, or maybe even with a lower resolution scan? And then one thing which quickly becomes important, certainly in the context of fMRI and certainly in the context of mesoscale fMRI, are the vessels. Because if you don't know where the vessels are or where they are, then you also don't know what your functional responses mean. So you got well, I was going to say high resolution, but I guess for vessel imaging, it's not even spectacularly high resolution. We've seen higher resolution, but for certainly these are beyond what we can access in fMRI, in the functional images at the moment. And then there's this interesting pattern, which is that when you project these onto the surface, the arteries follow a sort of, what do I call this, parasagittal path, whereas the veins seem to run in the folds, right? And then if you zoom in a bit, it's almost like you're creating a grid where the arteries go one way and the veins go the other. And there's some logic in that the supply of blood, when a brain is growing, the blood supply will organize itself to follow function. So when neurons fire a lot together, they get blood supplied together. It's a bit of a shortcut, but that might be the case. So perhaps these arteries are also, in a way, reflecting some of that functional organization that we are interested in targeting. And maybe this is actually a different route of getting to that mesoscale functional organization of the cerebellum. But so remember the veins, because this is also, they will show up in any fMRI data that you do of the cerebellum. So how do we get there? There's I had this nice figure also with the surfaces. A lot of the standard packages just don't do cerebellar surfaces, but I'd say if you care, then go check out Nikos's and Pierre-Louis Bazin's GitHub pages to see where they get these. And so if you have, again, high resolution, but not ridiculously high resolution, so these are 0.4 mil resolution amplitude ranges, then that is enough to create a surface which is good enough to project activation maps on. So, or good enough to inspect a T1 map in a complete 3D render. You can also see, so when it turns around, not all of these parts are sufficiently well done. So maybe we need to go back and revisit this. How do I get more SNR in my images? But we're getting there. And then what can we do? If we have this 2D space and we know what we're looking for in terms of mesoscale organization, where do we link this? And the interesting thing, or the thing that I find most fascinating about the cerebellum, is that it actually does everything. So it's part of every single major functional network of the brain, or every functional network in the brain has a hub in the cerebellum, and it's organized in patterns which are resemblant of the cerebral cortex. So there's a motor region which is roughly here. Here it's called saccades, but saccades are eye movements, so that could be part of a homunculus. There's a second homunculus running down, and there's this large part here, which is much more dedicated to cognition. So it's part of the default mode network, but there are also language areas there and things which are much more abstract. So by autobiographical recall is very, it's a very high level function. And then you can subdivide these in different levels. So the more fine-grained would be, say, individual digits, for example, but you can also look at intermediate scales. So there are a couple of very useful atlases if you want to venture here, which is made by the Jörn Diederichsen's lab. And I particularly like Caroline Nettekoven's one from 2 years ago now. So in terms of zooming in, and actually, because the atlases are great and useful, and I think will give us lots of useful insights, but they're not mesoscale. So if we want to go to the mesoscale, what are the ways in which we could target that? And actually, I did this image ages ago, but also because it's easy. If you do digit representations, also in M1. They're very clear, very binary also. I think you can give each finger a different color, and then you find this nice little rainbow. And you can extend this so that it follows. You can see more of these body maps. We did a version with eye movements and foot movements, and then some parts in between, and you see that there's an orderly pattern which is spanning that of where you would expect the motor zone from that large atlas to be. So is this mesoscale? Are they actually stripes? Are they the same stripes as what we think they are? So the first way I looked at this was just in the structure, in the T1, and because the myelination of these different stripes is slightly different. And if you squint, and I allowed that you have to squint a bit, then you see that these greenish boxes are greener than the blue boxes in between. So there might be a pattern, and that pattern is then in the same direction as where we see that functional gradient of the digits. So Nikos made this diagram of like where do we see the same sort of stripy thing in the brain function. But this, going back now to our vessel maps, these things are maybe a little bit similar. So if we want to really probe whether these are functional units or whether it's just the vasculature, then we need to do something a little bit better than only bolt-weighted imaging. But we've tried. Sorry, we're still trying. And one of the ways in which we do this is that first experiment of flexing and extending of the, well, not our forepaws, but fingers. And we know that there's this organization where we see the different digit representations lined up in an orderly manner. We also know the direction in which we expect the stripes, and so you can mentally sort of make a little 2D map of like movement type in one direction and digits in the other direction. And then if you do this, then you see indeed that so here we actually did flexing and extending and stroking, because stroking is a little bit more different even. And then you see that the stroking is more, I've said this so many times, more medial than this graph. That the stroking is more, more lateral than the motor task. So there's a step in terms of where we see these clusters in the like changing in between, if you look at only digit type or movement type in different directions. And we can so we felt that it's good that we see the difference between, say, stroking and the movements, but it's very hard to make these balance. So things like the flexing and the extending is so much harder that it becomes like it wipes everything out. So we thought we'd need something where we can balance two tasks a bit better in terms of their cognitive load. And then we looked at two motor tasks, which are more similar. So having two digits which you move, and the cues are very similar, it's just a color change here. And so you either ask someone to move the little finger on their left hand and the index finger on the right hand, or you ask them to like have these touch. And if I do it like this, then it's easy. But if I do it in the scanner where I can't see my hands, then it's actually a lot harder, and it depends a lot on my internal knowledge of where my hands are and how these are moving and where they would be touching in space. So that's it requires more proprioceptive information processing than the simple separate movement like this. And then when you do this, you get these, you can contrast these two tasks, and these you can then really look at in terms of where do we see the shift from one to the other, and where do these two within a cluster, where does the balance between these go? And if you look at that in the posterior, so the lower part of the cerebellum, then you see that there's a gradient here, medial to lateral, whereas in lobule 8 in lobule 5 it's medial to medial to lateral, whereas in lobule 8 it's more into the fissure. So the folding there is a little different. And here we didn't have the resolution necessarily to follow all of the cortex through, so it's not a true flat map, but we did generate surfaces or depths sort of over the entirety of the gray matter region. So we can, even with a task like this, you can make some inference about where these shifts are. And what I would also like to do is to move this into a more cognitive paradigms, and this is one which is not a mesoscale thing, but it is a very much a cognitive problem. It's much more related to aggression, where you're targeting a completely different part of the cerebellum. So we're this is a collaboration with the University of Utrecht, where somebody's studying very much the involvement of the cerebellum in aggression. Well, can we do this at 7T? Can we do this high resolution? Sure, let's go see what we can see. So she borrowed a paradigm from a cerebral study which didn't cover the cerebellum, where subjects are put in a scanner and they press buttons to gain points, and the more points they have at the end of the game, the more money they get when they go home. So they're pretty motivated, and the task is annoyingly straightforward. And then what happens is that at random intervals, points get taken away from them for no reason. And then they can retaliate, stealing points back, or not. So then you have two actions, which is the gaining money, but also the suffering and unfairness, which is like aggression to you. And because of the retaliation, you can look at active aggression. And then we see that there's a lot of activation, not so much in the motor areas, even though they are pressing the button, but the button presses are balanced, in these areas which are much more cognitive. So for the provocation, especially it's left lateralized, which it's right lateralized in the cerebrum, and in the for the stealing, it's on the right side. So if you compare this to those the atlases, then you can see that certainly for the stealing, there is activation here in this narrative language area, and in things like these networks which are in looking at maintenance and verbal fluency. So I think of this as people angrily muttering to themselves in the scanner while they go and get their points back. And I think these are also areas where we can find very small clusters which respond in a certain specific situation in a control feedback task, where hopefully one day we can also do mesoscale imaging. So another one which is much more cognitive is a task where we're looking at reciprocal behavior. So people working in a collaboration, and this is a task which is borrowed from a psychology pipeline, where people are being examined for whether they have features of autism spectrum disorder, and it's a controlled way of drawing together. And you can actually do this in the MRI. So here's one of my collaborators who's lying on his back, and this is a drawing pad, and this is a drawing which is made by switching turns between the black and the purple person in a planned way, which you can do in the MR, and then you can look at where do people engage during the motor task, which is obviously a lot of the motor networks, but you can also look at more of the sociable or social aspects of this task, and then again relate this to what do we see outside of this motor network, where we can look at what the more social networks in the cerebellum are doing. Okay, how are we doing for time? So I have 10 minutes. Of course, as I said at the beginning, we do in the end we do a lot of what we do in neuroscience also because we want to see what happens when things go wrong, whether that's neurological or psychiatric diseases. we set out to not just to study the system, but to understand the system, so that when there is fallout, we can say something about what goes wrong and how one might be able to solve it, or whether we want to do any diagnostic follow-ups or so. And one of the areas in which the cerebellum is particularly relevant is multiple sclerosis. As you probably know, MS is characterized by white matter lesions, and because they are everywhere throughout the brain, the problems of MS patients are also everywhere, as you can see on this nice map. And cerebellar lesions are particularly important to the disease, in that if you are diagnosed with MS and you at the time of diagnosis already have cerebellar lesions, then that is a very strong predictor of a more serious disease course, and it's also related to the sort of problems that people with really cerebellar disease also have, so ataxia, poor control, dysmetria. And still, and we've known this, I don't know, since forever, it feels, but the exact role of why this is the case, it's part of the diagnostic criteria. If you have cerebellar lesions, if you have a cerebellar lesion diagnosis, then you have MS. But we don't know why this is so important, or what that role is playing. And that's maybe because there's more to MS than lesions. So if you look at an MS brain and you just look at lesions and you can, well, these are the lesions, you don't need mesoscale or even 7T imaging to see these. But if you look at the cortex of an MS patient specifically, then you see that there's also an overall change in the gray matter. So if you look at the quantitative MR, and you, provided that your segmentation is sufficiently good, then you see that there is a local change there, which sets the patients apart from the controls. And maybe this is what's called, more than the lesions themselves, maybe this is what this volume loss is what's causing the cerebellar aspects of the MS patient symptoms. So we could see those surfaces. We wanted to see whether we could zoom in a little more, and then we went to acquire also 0.4 mil resolution images in MS patients, and then compared those to how things looked at the 3T. And there are actually a lot of things, a lot of features which you can see at this mesoscale resolution, which you're just completely unaware of at 3T, which actually makes the comparison quite challenging. So this lesion, for example, is kind of inexistent. It's maybe there if you look at the low resolution 7T image, but you can see much better where it is, and also how the tissue around it looks once you go submillimeter. And of course, you can try and look at the actual layer or the actual mesoscale features of this brain. So what we were interested in here is whether the layering patterns, or whether we would still be able to see the granular cell layer in the MS patients, and whether we would see it in the same way. So whether the iron-rich all little cells together, whether they would show up in a different way in the patients as they would in the controls. So we used here also a Laney segmentation of kind of a bigger block, with the idea of being able to make a laminar profile, not of the function here, but of the phase of these gradient echo images. And then there is so we do see a change in how steep that edge is that we're looking at here. So it kind of goes up and it goes down, and then when you average, there's always a difference between the patients and the controls. Whether this is it wouldn't be a diagnostic criteria, but I think it's good to know that these changes that we do see, that they are there at the mesoscale, and perhaps one day we will be able to learn something about these that will also tell us something about how this disease progresses. Okay, I'll do this very quickly. So another way of making samples small is by looking at individual patients. It's also something which mesoscale imaging general is good at. an individual to a patient, the individual is always important. We always want to have a diagnosis and then this one. So this is a person with a specific genetic point mutation, and then as a result of that mutation has ASD and very clear sensory motor problems. So just the way that he behaves. And because it's a point mutation, you can model this in a rodent, do segmentations of the cerebellum, and then look at where you see changes compared to controls. And then you can have a sort of a morphological confirmation of how good your rodent model is compared to your brain. Because you cannot have more patients, right? But you can have large groups of rodents, which you can then test about. And what works in the cerebellum segmentation-wise also works for the midbrain structures. We can do these in the rodents, and we can do these in the brain. So, ta-da. Yeah, that was my 40 minutes all done. In conclusion, apologies for not showing you any mesoscale fMRI of the cerebellum, but ask me back in a couple of years and maybe I will. But I hope I showed you that what we know what we're looking for. We're looking at these 3 different cell layers, and we know that at least 2 of those are MR visible, provided that we have enough resolution. We're at 70, very close to visualizing these properly. And we can do this both structurally and functionally. And with that, I'd like to thank my group and all the collaborators and you for your attention. Renzo, wait, let's stop the share.
Sorry, maybe Tyler can moderate the Q&A as it's getting noisier here.
Oh, you raised your hand. That's why
Sorry.
I did that.
Oh, sorry. I do have a question to start, though, about the PTX phase shimming. So I understand that it's easier to just do shimming instead of kind of going full PTX, but as soon as you do shimming, you could include the magnitude as well. Why did you focus on the phase? Was it just good enough and there was no...
Oh, no, actually, we did magnitude and phase shimming. We just didn't do like a Katie Points pulse where you modulate different shapes of different ways. But the optimization every time just wanted more power, like more, and then there just wasn't any point in we run all the elements at max, right? We run all of them at max power anyway. So there was never any point in downsizing any of the channels. So we asked Emma a few times to look at that, and then she did, and she said it wouldn't work, and then we tried again, and then it still didn't work. So I think there's not that much benefit in modifying the magnitude as well as the phase. But you could, maybe for a smaller ROI, there would be more of a point to that.
Do others have questions?
Can I ask a follow-up on the RF shipping stuff?
Sure.
Yeah.
Is this something that you do manually? Like you take a B1 map, you take it offline, segment it, put it into MATLAB, get 8 coefficients, then manually prescribe this, or like what's the workflow?
Well, so by now, I guess now that we've done this, yes, that is the workflow. But now what we do is simply select that one text file where we say cerebellar shim, load the cerebellar shim, and then scan. It's a Control-C, Control-V on a text file, which has also been scripted. We don't actually do any calculations for a normal fMRI session. We just use the universal offsets.
I see. So you scanned a bunch of people, you optimize jointly, and then you check those coefficients whenever you scan.
Yeah.
I see. Yeah, I'll let others ask more questions. I have a couple more things I can ask later.
Robbie?
Yeah, I mean, we use magnitude or historically have used magnitude and phase. Our amplifiers are not pegged, so maybe that is part of the difference. But It works pretty well. I mean, and it's completely automated. I mean, the MR tech uses it, and it just requires, you use your adjust volume in the Siemens,
Yeah.
environment. You use the adjust volume, and you shim over it, and it works. Um,
So do you have
90%.
any? Can I ask you something back then? Do you
Yeah,
have any
yeah.
feeling for how big the magnitude modulation is? Like how much are channels scaled up?
It can be at least 50% between transmit
Okay.
elements. So
Okay.
it depends what you're shimming on, right? So, if you're truly interested only in the cerebellum and you put your adjust volume and your shim volume over the cerebellum, um, then you get one shim. You get a very different shim if you shim the whole head, which,
Yeah.
if you want to do whole brain images and whole brain connectivity, then it is quite different. And the only way around that is to do dynamic PTX. So, full, KT spokes, or, we have a paper coming out with Omer Oran. Omer's now, he was our Siemens scientist here. Now he's at Stanford. That paper is accepted in principle, I think. But so we don't find universal pulses work as well. So we have, using some of the fast algorithms that have been developed by a number of the Siemens crew, as well as here. It takes 20, 30 seconds to optimize, a rare sequence, for example. So, all the spin echoes and everything. That works really well. At that point, you are at 3T homogeneity. And that makes, I think, a big difference. But my question to you was, you can make beautiful images at that point, but I still feel that motion is the biggest problem when you go to these high resolutions, because, if you move 300, 400 microns at 3T, big deal. It's a tenth of a voxel, or a third of a voxel. But if you move 300 microns at 7T with some of the beautiful images you're showing, you're in the next voxel. it's a big problem. I mean, what do you see as the potential solutions there?
So for us, the
Practical
FatNav based,
solution, right?
right? So for us, that prospective motion correction is actually pretty good. I think.
Okay. I mean, looked really good.
Yeah. Yeah. No, it does. Like, but I know that it doesn't. So I'm not sure if it's the best solution. I just haven't done enough prospective motion comparisons to tell you that. I think there might be ways, and so this costs about 400 milliseconds or so to acquire the volume. It's also, when you do motion correction, you can sort of correct up to like a tenth of your resolution. So we acquire the FatNavs at 2mm resolution, which means that it should hold until 200 microns,
Right.
which is enough. But that means that, like, then you have to have a lot of faith in that factor 10 step, right? So we're not at 200 microns yet. So I think for now the FatNavs work, but there are other options out there which might also work, and which might be able to push things a little bit further in terms of the precision and being able to target resolutions beyond that.
So you're using a FatNav, so you can use this in sort of the T1 recovery period and not bother the proton ionization.
Yeah.
Basically. I mean, we could do that with rare too, I suppose, because we have a big, long TR.
Yeah, you have that time
for SAR purposes.
Yeah, and we run the FatNavs at the super low flip angle, so in terms of SAR, it's like it's invisible.
Yeah.
There's
I mean, it looks really good.
There's enough in the skull that it would work. Yeah, that is, I think, like, it works. So we kept using it, and then I don't know, move on to the neuroscience, right? Like that. So I see this is one of the things where I kind of go around in circles, and like I need more SNR. So then I look at my sequences a bit more critically. Then I get more SNR. Then I have to look at the motion. Then I do neuroscience, and then I think of something else that I would want to do neuroscientifically, and then I have to go back and actually I don't know, maybe consider all of the assumptions that I started out with. And should I maybe still go back to the coil? Should I don't know. But you do it in a coil, so we're good.
Are there other questions? If not, I have kind of a little bit of a follow-up of that. I mean, you mentioned a little bit about the dead time and how it's a little bit convenient to put that navs into things like your MP2 rages, but I was curious, I didn't quite catch it from the slides. Are you doing perspective in the functional scans that you're doing right now, or are you leaving that until you need it?
So we did this, well, not in the Cerbellum, but we ran this acquisitions with the line scanning, where you, instead of acquiring a slice, you just acquire a line, and then you get super high resolution along that line. And then, well, we kept also having questions from the reviewers about how do you know that people don't move? Because you can't see it, right? So then we put the fat navs in there as well, because then you can see whether people have moved. But it's such an interruption of what you're doing, and then you really have to think of how do I keep the magnetization stable? Because if you interrupt your sequence like that, then you get to, I mean, so Ravi said it the positive way around. You're not interfering with the T1 recovery when you're doing a fat image, but when you're actually continuously scanning, then you do want to, like, you want to keep your you want to keep that T1 weighting constant, right?
Yeah.
So then it's not fat navs, but water navigators of some sort. But then, like, there's the undersampling is a bit different and stuff. So I think there are ways in which you could do this in the fMRI, and it would be it would be a good idea for anything which takes a bit longer, right? Like once you get to, if you make the resolution really high and you have like large coverage and you get a TR of, I don't know, 2.5, 3 seconds, maybe it's not so bad to then have another half a second dedicated to actually having prospective motion correction. But we do a lot of the acquisitions that are whole brain. We do them with 3D APIs, and then like motion, just the normal motion correction also does a lot. It just doesn't maybe cover it wouldn't cover the within volume movement. But that
makes sense. Yeah.
Yeah, on the whole, like once you have a task in there and people are actually doing things, then I think the within volume movement is not always the worst problem. But you could look at completely different things, right? You could do I don't know, spirals.
For sure. If there are any other questions, please interrupt me, but otherwise I'll just keep asking questions too. I was curious to go back to the RF shimming a little bit. It looks like from your coil that in the case of the Nova coil, obviously we kind of only have our degrees of freedom going around the head. We don't really have the up and down. And I was curious, does that kind of mean that all of your solutions that you're doing are just making it so the back of the head is a little bit better, so you could potentially get visual cortex in these as well?
Yep.
Or are you kind of is there something I'm missing that's giving you the Z direction?
No, I think both. So the way that the brain is, I don't know, asymmetric, I guess, once you position it in the coil, there is so we see this signal, or the lobe B1 areas in the right posterior lobe of the cerebellum, and it's not constant in the Z direction, right? So in V1, if you do like in the quadrature mode, there is actually signal in the right, occipital lobe.
Mm-hmm.
So there is some variation in Z, which once you start shimming also, so the degrees of freedom might only be like in XY, but once you say, oh, I want to focus my signal here, then there is also a change in how far down the B1 is acceptable. Mm-hmm. If that makes sense. I've seen somebody do something similar also with like the carotids, that if you shim on the carotids, then you can also make the P1 of the nova coil go down and not actually have a proper coil, like a different coil set in the Z direction.
Very cool. Other questions? Okay. Renzo?
Yeah, Renzo, go ahead.
I was going to ask about the vessel segmentation in this very nice slide where you showed that the arteries are within the ridges and the veins are kind of perpendicular to that. How did you quantify that? Was that kind of manually going in, or do you have a cool vessel segmentation tool there as well on top of the? No.
Yeah. Thijs the book had a cool vessel segmentation thing. Don't ask me what it's called. I can find out for you. No, it's not a manual segmentation. There's a no, pass. There were different versions. I'm not going to say which one I think it was in the end.
And I ask about the fat nerves, like how long they take and how long does the feedback require processing and updating of FOVs? And do you have enough time in your sequence to?
They are roughly 400 milliseconds long. And the update period is, so you could do this differently. So I think it's half a second. So you actually need a second, which is fine in the MP2 ages, but anything else it's painful. But you can also update with a lag so that you have, like, if you don't have time, then you can, like, say, update back previous on the like on the difference between -1 and -2.
And when you put this inside the GRE sequence, then you have to pause your sequence for a few TRs, or?
Yeah, so then you have to think of where you want it, right? And also on the Philips system, the GREs are grouped anyway, so you can say I want one per K-space plane, for example. I think that's what we usually do. So not per shot, right? Because per shot that would be silly. But kind of like the way the MP2 ages are implemented on the Siemens systems. You do a K-space plane, and then you have a break, and then in that break you scan, and that way you don't create any funny patterns of T1 weighting in K-space. Because if you do it, I don't know, you have to think of where you want these breaks, right? So that any variation in the contrast weighting has to be like smooth throughout the K-space.
So do you put dummies to kind of transition?
Nope.
You don't need dummies?
No, you don't need this. I think that would actually take too long if you would start, because then you would need, you do a fat nav and then you have a pause and then you do dummies and then you continue. I think then the interruption becomes like kind of the size of where you want the temporal resolution of the fat navs to be. So we can choose, like with the fMRI fat nav implementation, I think we did every 30 seconds just so that we could check whether people were drifting out of the coil. If you want it to be much, if you want the temporal resolution to be shorter, then you have to also be more worried about how long that interruption is and how much information you can get out in that time.
If you stop to acquire your fat nav for like 400 milliseconds and don't keep hitting with your alpha pulses, don't you need to go back to steady state again? A couple TRs of.
Yeah, but so if you think of it as a 3D space, if you make sure that you have a T1 weighting over your K-space, but you have it in every plane in the same way, then it doesn't create any.
It is broken steady state, but broken in the same way. So it's
yeah,
that makes sense. Okay, thank you.
Very cool. So we are a few minutes past the hour. I assume you would welcome follow-ups, but I think for today,
sure,
we should move on to other meetings. Thank you so much. This was really cool.
Thank you.
And it's
Thanks
like a very nice body of work towards being able to like do this consistently. So